Namaste students. Today I am going to discuss about the topic genetics of immunoglobulin. It means we are going to discuss about the gene organization of the antibodies. So as we know that antibody has heavy chain and the light chain, and they have variable region and the constant region. The constant region is fixed in each antibody. The variation is found in the variable region. So that variable region will decide which kind of antibody will be there, which kind of antibody will be there for the identification of the antigen. So there are millions of the antigens can be present in the environment, and for those millions of antigen, for identification of those millions of antigens, we need millions of antibodies. So how can we manage, and how the body organization will manage for the identification of those antibody, those antigens, because they are very large in number. So how this can be done for the identification of the antigens? So there is a very complication situation. So the problem can be resolved by the variable regions, because constant region is very fixed, but the variation is found only in the variable region of the antibody. So only the variable region will change its structure, its genetic organization for the construction of complete antibody. So first we are going to discuss about the light chain of the antibody. So in the light chain, there are two parts, kappa and the lambda. Kappa light chain in the lambda light chain. In the kappa light chain, as we are observing here, the light chain is composed of the variable fragment of the joining J segment and V segment. They are V segment and the J segment. So V segment are in the range of 1 to 41, 1 to 40. So this is the double stranded DNA, and this is the light chain part. In this light chain part, there are two regions. First one is V segment and the second one is J segments. So only these segments will decide the which kind of antibody will be there. So in the V, first we'll talk about the V segment of the kappa chain. They are in 14 number. So V1, V2, V3, V4, V5, V6, V7, V8, V9, V10, 11, up to 40, V40. Similarly for the J, it will be for the 1 to 5. Okay, J1, J kappa 1, J kappa 2, J kappa 3, J kappa 4, J kappa 5. And then this is the constant region. We'll start here. Okay, so suppose that the antibody will be genetically diverse. How? Suppose here in this light chain, in this kappa chain, we have V1, and this V1 will be next followed by this J chain, and suppose only JK1 will be here, which will decide the genetic organization of the kappa chain. Now we talk about the genetic organization of lambda chain. In the uh, kappa chain, there were two sides, variable region and the J region. J for joining and V for variable. In lambda locus, it is found in the chromosome number 22 in the human, and it is composed of four uh, carbon region, constant region genes. These are four constant region genes, and the region of V1, V and the region of J will mix together to make a variable region, complete variable region, and this J region will uh, be used for the joining of a C region. Okay, this is for the joining of constant region to the join variable region. Okay, so now this is the arrangement how the the genetic organization of a double of a antibody will be arranged in the double stranded DNA. Suppose this is the light chain of the kappa, and there are V1, V2, V3, and suppose V1 region is selected. Okay, here are G1 to J5. So in the light chain here, one V1 region, there is a cut between the V2 and the Vn, which will cleave this region completely. Okay, V1, V2 and Vn, there will be a cut which will cleave another region. So here we can find here that V1 that is joined by V2 V2, and then this J4, J1 and J3 will be cleaved here, making it J4 to J5 complex. Then this V1 will be cleaved here, okay, making it V2 and J4 J5 complex, which is combined to constant region. Here this V2 and Vj will mix together and cleaving this J5 region, making it a light chain complex of V2, J4 and constant region. This is the complete antibody of the kappa chain. And to this region, the translation will happen for the construction of the complete antibody. Now we are talking about the heavy chain region. In the heavy chain, there are three portions because it is heavy chain, so definitely will be larger than the light chain. In the heavy chain, there are three regions: V region, D region, and the J region. V for variable, D for diversity, and J for joining. It J is joining the D region to the constant region. Okay, so how the VDJ recombination will happen? In the V region, there will be V1 to Vn, n number of portion will be there. And in the D region, there will be 20 approximately number V, D1, D2, D3, D4, D5, D6. Okay. In the J region, G1 to J6 will be present. Now talking about the VDG recombination in the heavy chain. So suppose there is a V portion which contains the V1, V2, V3, V4, V5, V6 here. Okay, V1, V2, V3, V4, V5, and in the D1, D1, D2, D3, D4, D4 up to 20, and in the J1. So from these V1, V, D, and J, only one single portion will be there. Only one single portion will be there. Suppose V1 will be there, D1 will be there, and J1 mixed together to constant region, making a complete heavy chain. So how the recombination occurs? First, what happens? The D portion and the J portion will mix together. So first recombination will occurs between the D region and the J region. So suppose there the D region, in the D region, the there will be cleavage between the D1 and the D20. So this portion will be completely cleaved out, making it only D portion, D1 portion. So this D1 portion is here, and in the next J1, J2, J3, J4 is not required, so they will be cleaved, making it a J5, J6 complex. Okay, so J5 and J6 is here. After that, V J5 and J6 combinations, VDJ recombinations will be there. So how the VDJ combination will be there? First, here we can see here V1, V2 is there, and D1, J5 and J6 are combined to each other to the concentration. This D1 is here, this J5, J6 is here, and this V region. Okay, here on this V region, V2, there is a cut between here V2 up to Vn. Okay, from this V2 up to Vn, approximately 14 number. Okay, so there is a cut. So all the portion from V3 to V40 will be cleaved out, making it only V1 to V2, D1, D1, J1, J5. Okay, after that, this J1 portion will also be cleaved here. Okay, this J1 portion is not required, so it will be also cleaved here by the lytic enzymes. This lytic enzymes will cleave this portion, and the DNA ligase will join only V2, D1 and J5 portion here and D6 portion here. After that, this J1 portion will be cleaved from this side to this side, making it a complete H chain, heavy chain. So by this method, the VDG recombination will be there for the making of the complete arrangement. So this is the complete mechanism by which VDG recombination will occur here. Now we are talking about the class switching. What is the class switching? Class switching is a method by which type of antibody will be selected. Means we are selecting which type of antibody. Either it will be antibody G, IgG G, IgG A, IgG E, IgG D, or IgE. So immunoglobulin E. So which antibody will be selected? So when the variable recombinations happened here, this VDJ recombination, this class H will be here, in which these are the chains: mu chains, delta chains, gamma 3, gamma 1 chains, and alpha 2 chains. So mu is responsible for the immunoglobulin M, delta is for that immunoglobulin D, gamma is for the immunoglobulin G, and alpha is for the alpha, immunoglobulin A. On the basis of the presence of these antibody, on the presence of these chains, the antibody will be selected. So how they are selected? Mu for M, delta for D, uh, gamma for the G, and alpha for the antibody A. So when these will be selected, they are making a particular kind of antibody. So how they are selected? So here I'm going to explain it. Suppose that we need immunoglobulin, suppose the body need immunoglobulin alpha, immunoglobulin A. So what will happen first? The first chain is for immunoglobulin mu chain and delta chain, gamma chain. So if we want alpha chain for the which is responsible for the immunoglobulin A, then there will be a cleavage between this side to the this side. Okay, from this side to this side. After cleavage to this side, this portion will be removed here. Okay, this portion will be removed, making it V2, V1, J5, J6, uh, gamma 1 and alpha 2. After that, this gamma portion will be cleaved here, and the next antibody will be alpha, which will make it the immunoglobulin A. The first antibody after the J chain will be the antibody that will be synthesized after the VDJ recombination and the class switching. So this portion is responsible for the variable region, and this portion is responsible for the constant region, which will decide the type of antibody in the heavy chain. And it will be dependent on these presence of these chains. If we require the immunoglobulin M, okay, then what will happen? There will be cleavage from this portion to this portion, making it only immunoglobulin M, mu chain, making it immunoglobulin M. So this is the procedure by which the immunoglobulin will be designed. So our topic is over here. Thank you very much.